Hello.
Rachel,
Hello.
how are you doing?
I'm doing okay. I'm gonna try to log in with two screens so I can show my PowerPoint, but then blip between like other things.
That's totally fine. I just let in Rachel number 2.
Yeah, there we go. All right.
Are you doing it from the same Zoom account or you have
No,
two?
I made another Zoom
Yeah.
account with
For a
my
little while, I had that going when I was teaching, and I can't remember why, but it ended up confusing me. But that was for a different reason. So yeah, I'm really looking forward to this.
I'm excited. I'm excited. I'm looking.
And we may still have a couple people joining us here in the next couple of minutes, but I wanted to welcome everybody. Those of you who don't know me, I'm John Wares. I'm a professor in ecology and genetics, but I'm also a curator with the Georgia Museum of Natural History and the current president of the Friends of the Georgia Museum of Natural History. And the Friends started this talk series so that we could sort of appreciate our biodiversity, natural history, and museum adjacent research at the University of Georgia and beyond. So even though we're not doing this in person this year, it's something that I think really helps bring together our museum community as well as the friends of the museum. So I wanted to make sure and remind everybody that if you go to gmnhfriends.org, that's the organization that supports this series and in many ways supports the museum itself. And so we're hoping to keep this talk series going into the next year, but remember that your support, your donations, and your membership help us tremendously. And so with that, I'm super stoked that this month I managed to twist the arm of one of the talented students who I serve on her doctoral committee. So Rachel Perez-Udell is in the Department of Plant Biology. She's a doctoral student. Good morning, and so she's going to be telling us about the work she does on Geranium maculatum, but with a really interesting twist that a lot of you may find yourself generating new data for her in the next year. So with that, Rachel, I will let you take over.
Hey y'all, so you might notice I have two screens on this, and that's because I'm going to be doing a little bit of jumping between one visualization and another. So to begin with, I want to say that I'm going to try to make this talk as interactive as possible to kind of emulate the vibe that we would get if we were actually at the turtle pond. So I've got this really cool website called Poll Everywhere that I'll be using to get kind of clicker responses from you to get some engagement in the talk. So I've put a link here. It's also, if you look at the chat, there's a link to a Google Doc, and that Google Doc has the link to the Poll Everywhere, as well as some hyperlinks to the various sources and cool iNaturalist projects that I'm going to show you. So if you could go to this URL, you could do it on your phone, on your computer. You can also text it to your phone. I'll leave this up for just a few moments so everybody can get that down. Then I will, oh, I see something in the chat. Oh yes, okay, so John put it in there. So I'm gonna go ahead and stop sharing here and flip over to my other screen because technology. So my story for you guys today is really going to be about color. So I want to begin with, and this is just to make sure that everything's working right, with what's your favorite color? All right, we've got one response. We've got black, and I'm pretty sure that was my husband putting that in. Green. And the link is also up here as well. All right, we're getting some more. I don't think I can respond to my own. All right, well, we will keep this up for the chat because I'll have other questions for you. But for now, I'll go ahead and move on. All right. Come on, PowerPoint. All right, so today I'd like to tell you a story about color. color, it's everywhere in nature. We see it in the lovely peacock's feather as he searches for his peahen mate. We see it in the wide diversity of flowers on this earth. We've even noticed that certain colors can entice certain pollinators, part of what is known as a pollination syndrome. It's why bee-pollinated plants are so bright in color and why bat and moth-pollinated plants are white. We see it, or don't, in camouflage, as well as in mimicry. So if you look like the thing that tastes bad, you likely won't get eaten. And we humans have even brought many animals to near extinction, like these lovely mollusks here, to create dyes such as Tyrannian purple. Fun fact, it's super expensive, and that's why we have this connection of purple with royalty, because they were the only ones that could afford it. Come on. All right. And just to set the stage, I'll talk about two really well-known examples in the biological world. The first is the peppered moth. Everybody was probably told this story in their first year of their introduction to biology class. Peppered moths usually blend into the bark of their trees, of their surrounding. So typically the bark would be white, and this guy would blend in really well. But we noticed that in areas where the Industrial Revolution happened, soot covered these trees, and this darker morph became more prominent. So you actually had the shifting of like where the soot was covering the trees, the blacker morphs were, being camouflaged and they weren't getting picked off by birds. Another really well-known example, at least in theory, is sand blossoms, or Linanthus prairieae. This species comes in two colors, either blue or purple, depending on what you want to consider that color, or white. It was the seemingly random distribution of color that led famous geneticist Sewell Wright to his isolation by distance metric, which is a commonly used metric in population genetic studies. So I'd like to hear your thoughts. First, to name a species that exhibits variation in color. And now I'm going to do my fun flipping because I have it as, haha. So any species, animal, plant, insect that exhibits some type of variation in color. Maybe the variation is during the life cycle, or you can yell it out.
Do we have to reload that page?
I don't think so. It should. Does this not show up for you when you go to it?
I'm still seeing what's your favorite color.
Okay, hold on. Oh, maybe I need to click this. Does that work?
Let's see. Oh, how weird.
Well, then maybe we'll just trash this. All right, then. We're just going to trash this idea. It was great while it lasted, which was all of negative seconds.
I actually got to it eventually, but it may be that other people are having problems too, so.
Okay, I was doing it because I was like, maybe people don't want to shout out, and then they could just type, and then you don't know what response you gave.
Right.
But anyway, name a species that exhibits variation in color. And you can post in the chat too. Ah, there is chat stuff. Spiny back spider. I do not know what that is. It sounds cute though. Butterflies. Yeah, butterflies do. I was also thinking deer this morning because fawns are, they have the white like speckling in their back and then they grow up and they're brown. So that's kind of a variation. It's more of like a variation in like time scale. All right. And then, oh, there's another chat. Sorry, the chats are hard for me to like. Labrador retrievers, yeah. We've got a chocolate lab named Scout. He's a glutton. Don't give him food. Why do you think it exhibits variation in color? So it could be very simple, like with the Labrador retriever, it's, we bred them, we continuously like breed them, selectively to be like, we want to get brown labs, we want to get black labs, we want to get yellow labs. But there are tons of reasons why you may show variation in color, going back to the first slide, it may be, some type of camouflage. There could be some natural selection towards it. It could be random drift. So that brings me to my species and kind of like what we're trying to figure out about Geranium maculatum. So I'd like to introduce you to my study species where I investigate color variation in petal and pollen. It's simply a beautiful plant with these gorgeous blooms. You may know its relative, Pelargonium. It's the geraniums that nurseries tend to sell for gardens, so you can consider that its cousin. It's a spring ephemeral, meaning that it blooms in the spring and disappears later on in the year. So if you were to look like in winter, the leaves would probably be dead and it would just be a rhizome under the ground. It's found mostly in temperate forests in mid to eastern US. So I have this map here on all these bright green spaces or counties where it's natively found to exist. It can bloom as early as March and as late as June, but we've noticed that its peak season happens to be around April and May, depending on where the population is located. So southern populations tend to bloom earlier than northern populations. It's pollinated by a variety of small insects, including bees and butterflies. This fact is beyond the scope of my research, but it's important to note that not all Geranium maculatum actually have pollen, and that's because this plant is gynodioecious, meaning that some of the plants are hermaphrodites, meaning they're both male and female, while other plants are just female. So we have no male plants, like male-only plants, either hermaphrodite or female. So I've introduced you to the plant, and now I want to introduce you to the color variation within the plant. So Geranium maculatum displays a wide variation in petal color and pollen color. Up on the top, I have kind of a swatch of the various petal colors we have. So they range from this really like pale powdery pink to this like deep purple. And we typically consider these two pinks to be light, the middle pink to kind of be a darker pink, and these last two to be darker. So like the darker morphology. The pollen, however, is a little bit harder to ID in the wild because it's more ephemeral than the petals. After all, these pollen-bearing plants are hermaphrodites, and after they shed their pollen, they become female. So they don't always bear pollen. So I have photos here showing most of our pollen variation. We start off with this kind of like really neon-y yellow color, followed by this creamy diluted yellow. Then what we consider like a light purple and a dark purple. So now to set up the story, the foundation of the story, there's this variation in color, and what Shumei, my advisor, and previous graduate students in the lab realized, they were really interested in studying gynodioece. So they were doing field work to survey population, seeing the percentage of females to hermaphrodites. And this is because gynodioece is considered to be the stepping stone between having one sex, so being hermaphrodite, and having two separate sexes, so having like female flowers and male flowers separately. During their fieldwork, they took a few notes about the overall color of the population, and I used these notes to construct a map of like their lower field research area where they'd really kind of taken these notes. And based off these observations, we found that these darker petaled individuals were found higher in the mountains, higher elevation, and these lighter petaled individuals and kind of lighter pollen individuals were found in these lower elevations around Athens. So if you were to pop by Lake Herrick or the Orange Trail off the Botanical Gardens or Bear Hollow Zoo, you would find these pale pink geranium with yellow pollen. So we believe that like if it was possible that elevation and color in the species were related. So we kind of wanted to test this out. And so that led to kind of the foundation of my dissertation. And one of the questions that kind of started off this dissertation was, is there a geographic pattern to the floral pigmentation in Geranium maculatum? I do have some answers for you in this talk. However, we realized that we have this really large area to sample. So I'll point you back to this map of geranium. So that's a lot of ground for a singular graduate student. To compound this issue, these plants have a really short bloom season. And COVID destroyed my ability to sample last year. So, you know, within like a window of a dissertation, I haven't had a tremendous amount of opportunity to go out. So I'm hoping this year I can finish my actual fieldwork. Then of course, there are just the perils of fieldwork in general. I remember this very distinctively. I was in Virginia. I was with my husband and my dog. We were in the car. We were driving up to this field site that I thought I found, and it was just pouring rain. It was absolutely miserable. And I'm trying to find these geranium in this park. It's muddy, it's rainy. I've like put like a human poncho on my dog and tried to like strap it down on her to like keep her as dry as possible. And she's looking at me like, why the heck did you bring me here, Mom? Like, you stuck me in the car and we drove all day, and like, this is what I get. This is miserable. And I couldn't get an accurate, you know, count of the pollen color because the rain just washed it away. And I was like, oh, okay, there has to be a better way to get, a creative way to get the data that I need. So I thought to myself, citizen science. I could aggregate other people's time in nature to crowdsource my data. So for a question, I have, what is citizen science? A small discussion, AKA I'm tossing y'all into breakout rooms. I also don't know how to put people into breakout rooms. Oops, I didn't want to give you the answer. Actually, how many people are in the talk right now? We don't have that many, so we can kind of just make it like one breakout room, which is just us, if that's okay with everyone. So what is citizen science?
I've heard of it, but I'm not going to give the answer.
Well, I think it must be data collection by people who are not actually involved in science.
Great. Does anybody have anything that they want to add to that? Okay, well, I have, and I've given you guys a link too, but that's basically what it is. It's just participation, public participation with scientific research to help increase scientific knowledge. So I know this is a pause to my story, but I wanted to stop to highlight how cool and useful it is. So I thought that this was a good topic since we're getting into hummingbird season. So the ruby-throated hummingbirds are the lovely little hummingbirds that we'll get through our area very soon. So set out your hummingbird feeders. And there are actually multiple citizen science projects that are dedicated to tracking their migration throughout the US. And so what they do is they usually set up these websites asking people to record occurrences. And this is the 2020 map. And it allows us to get this idea of when we should be seeing these hummingbirds in our areas. And if we are able to aggregate multiple years of data, we can see how different external factors such as like climate change could, you know, influence or impact this migration pattern. So if you look at where we are, you know, we're getting in the month of March, so they should be coming up soon. So then, how do we as plant people get citizen science data? So traditionally, for us, if we wanted to find a wide source of data regarding plant occurrences or morphology, we would use a herbarium. And a herbarium is a collection of dried plant specimens, and all are labeled with certain pertinent information. We here at UGA are super fortunate to have a very large and awesome herbarium. It's located in the Miller Plant Science Building. We're even more lucky to be in the digital age because many herbarium have actually begun to digitize their collections, making them easily accessible to everyone. So let's look really quickly at what a typical herbarium specimen would look like in terms of information. So what I have here is actually one of my herbarium sheets that I did in undergraduate. You'll see that there are 3 pressed plants. The flowers have been, at least this flower has been dissected and spread open, so you can see the inner workings, while others have been left intact. And this one was just kind of cleaned down the middle. So what information somebody would see if they were to pull the sheet would be the species and a family name. And this of course can change with time because we do sometimes change species names, especially if we realize that 2 species are one. Next would be location. So you'll notice here that I got this in Lumpkin County. There would be coordinates. I gave the elevation. I also gave some verbal instructions. So it's in Cane Creek at Radar Ridge. I gave some information about like what the area looked like and kind of what the conditions were like. I noted what color the blooms were, and you may have noticed that they no longer look that color. That's important. Then I'll give you the name of the collector and the number and the collection. So this was my 25th herbarium press and the day that it was collected, which was October 20th in 2015. So they're super great sources of knowledge. However, for my purpose, they don't retain color really well. So that's obviously a problem about asking about color across geographic area. Additionally, you can't just go around plucking random plants from areas. People tend not to enjoy that. So you need a permit. So it kind of like restricts access to those people who either are collecting on their own property or have gone through some type of like permitting process, or at least like some verbal agreement that they could take a specimen. However, no one is stopping you from going out and taking a picture of nature. That is, of course, as long as the nature isn't trying to attack you. So maybe there are some dangerous parts of nature like bears that may be harder to take pictures of than flowers, as flowers won't maul you, depending if it's a dangerous plant, you know, you don't want to hug the poison ivy. So how am I then going to get my data if I can't use traditional herbariums? Well, there is this really cool website called iNaturalist, and I absolutely adore it. Here are just the stats from 2020. So if you think that just in 1 year, they got over 22 million observations of almost 200,000 species, then there's sure to be records of my plant, which is considered common. It's common wild geranium. So I want to take an opportunity to just show you guys what a typical iNatural listing would look like. Let me share on here. Does anybody have a particular species that they want me to look at for this demonstration?
Marbled salamander.
Okay. All right, marbled salamander. So what's really cool about this is that you don't have to be a scientist to use it. As you noticed, I input the common name. So you can search by common name. You can also search by scientific name. So let's just click on one of these records. So here we have a picture of the little guy. He's actually really cute. Look at that. It gives you when it was observed, when it was submitted. It gives you location. So this one was in Wayne National Forest. If you look closer in details, you can get lat long, as well as how accurate those coordinates are. You've got the community's curation of the specimen. So what's additionally cool is that you don't need to know what you're submitting. If you don't know, the community will come together and suggest an ID. So if enough people suggest an ID and the location is good, then it gets a research grade tag, and I'll talk about that later. There are other things that you can do as a community. So I can say like, hey, this guy is definitely alive. And people can agree on that. I don't know the life stage, so I would leave that blank, and I don't know the sex of it. But you can also people can also give extra information. So sometimes when you look at where it is, people will like write things like, hey, if you go to this sign and turn right. So in terms of like data, it's very similar to a herbarium listing. But what's cool for me is that it retains its color, which is the most important part. All right. So then let's take a look at my species on iNaturalist. I've got a few examples of some of the occurrence records. And as of last Thursday, there were at least 15,000 occurrences. So what I did to collect my data was I used GBIF. And basically what GBIF is, it's where iNaturalist and other citizen science websites funnel their research grade Creative Commons licensed occurrence, meaning that like I can use these occurrences for research and use the photographs. They also give you a really great citation. So I could share the citation in papers or with other scientists, and they could have the same data set that I was working with. I took my data set and I further restricted it by coordinate uncertainty. So usually phones are really good at getting GPS location, but sometimes they're not. So sometimes you might get an occurrence where it's like, yeah, there's this geranium in Georgia, but the coordinates uncertainty is that it's like somewhere in Georgia, not just like somewhere in a park, but like somewhere in Georgia. So sometimes you kind of have to make sure that you're getting what you want out of it. And I got this large CSV file with about like 6,000 records that include lat long, a link to the occurrence record, who did it, the state that it's in, any additional information that they had, like, hey, it's by a waterfall. So it's really useful. So this was my data collection pipeline. So like after we got the CSV data downloaded, like what did we actually do with it? Like how did we take these 6,000 records and get color from it? Because I was not going to shift through 6,000 photographs. That wasn't happening. So I actually had a collaborator on this project, and this project was born from that horrible trip in Virginia, because I looked at my collaborator and I was like, there has to be a better way to do this. I have an idea. You said you wanted to learn coding, so like, let's get to it. So I'm going to quickly walk you through our workflow. So we first, got that CSV download. So our first hurdle, we had a few hurdles with this. Our first hurdle was to toss out any photograph that didn't contain flowers. As much as our lab really loves flowers because we're the reproductive lab, we've realized this is but a small portion in the life cycle of this plant. So there are actually a lot of observations that are just leaves or fruits. And that's not useful for me either. Then our second hurdle was to get an accurate sense of the color for just the petals. We don't care about the rest of the image. So we started this off by just getting HSV values for the entire photograph, so like each pixel. And so HSV values, just computer speak for dimensions of color space. So hue just being like the base color. So like, is it blue? Is it red? Is it green? Saturation being like how much of that color is present. And then value being like how much light is in the photograph. So it could be like really, really dark, almost black, or like really, really light, almost white. So then after we got these HSV values for each pixel, we did this k-means clustering within a photograph in an attempt to cluster light colors with light colors. Our goal was to end up with something over here where we could pull out just the petals. So our goal was to like get the algorithm to like cluster all the pixels that were like within the pink purple color space and ignore everything else. So this bottom photograph is just highlighting everything within that cluster, and everything else is, tossed aside. Okay, so that's like one hurdle. Like we've got an accurate, idea of like what that petal is, but now how do we like determine like everything? So then we did the same clustering technique to cluster like petals with like petals, which led us to 3 bins of observations, light, medium, and dark. So basically we took all the photographs and they were represented just by the cluster of their petals. And we said, hey, cluster like with like. So everything that's kind of like in this light area of the color space should congregate together. Everything that's darker should congregate together. And then we tackled the records without flowers by simply tossing out any photograph that didn't have a pink or purple cluster. So like if this came out black, then like we deemed that there wasn't a petal in it. So it was, tossed away. So I first wanted to show you like the lower appellations, because I started this talk talking about the lower appellations and how we thought elevation and petal color correlated. So right here, I have this map of Georgia, South Carolina, North Carolina, Tennessee, and I've got 3 different colored dots. The really light dots represent that light cluster. These kind of fuchsia magenta dots represent the medium class, and then these plum dots represent the dark class. And if we look like closely at this, we notice like, so this reddish brownish area is higher in elevation than the green. And we notice like, okay, there's darker petals in there. And then if we like step back and look at like this slightly less higher elevation, it's like medium. Sure, there's some pink in there, but for the most part, the pink kind of go to this lower elevation. So like maybe we've got something going there, just maybe, which led to a project that my undergraduates, Mackenzie and Emma, worked on. And that was just looking at kind of the lower appellations. And we've noticed through literature that elevation will correlate to UV radiation and temperature. And these increases in elevation have been known to increase concentrations of anthocyanins in plants. So anthocyanins, which are likely the pigment underlying the medium color, can confer some benefits. You can consider it like plant sunscreen. So first, let's consider just UV and UVB specifically. So UVB is that wavelength of UV that does a lot of damage. It's what causes sunburn on us. And likewise, it can damage plant DNA too. So anthocyanins, especially darker ones, so these like darker purple pigments, act as plant sunscreen against that DNA damage through absorbing the labelings. Likewise, there have been some recent work that suggests that this absorption can actually increase flower temperature and aid with reproductive success because it like warms up the sexual organs. Anthocyanins can also increase tolerance to cold temperatures. So this project's still in the works. We need another data set, but they've been looking at the effects of temperature and UVB on pollen viability in Geranium maculatum. So kind of switching over to, well, we want to expand it to look at like both petals and pollen combined, but that's just kind of something in the works that relates to that. So now let's look back at kind of all of the US. So same 3 color dots representing light, medium, and dark. And we kind of noticed that like, okay, you know, when we step back, the map looks a little bit more messy. Sure, there's this like high congregation of dark in the Southern Appalachians, which is like the highest point of the Appalachian Mountains. But then as we move north, it's kind of just everywhere. Like there's not really, there doesn't appear to be any rhyme or reason to petal color up here. But then over here by this like New Jersey, New York coastline, it seems to be majorly pale. So we're really curious, like, well, is there anything that could explain this? Maybe it's just random, it's stochasticity. But I'll just show you guys a little bit of my data analysis that I've done on this. So we were like wondering like if there were any factors that could influence petal color. So I ran a random forest model, and that's just fancy. I guess we can just consider that like a fancy data interpretation mechanism to just look at how various bioclimatic variables could predict petal color. So the two that really came out were elevation and world climb variable 3. So this is actually this data set that's put out for public use. It has 19 climatic variables, and variable 3 is isothermality, which is the mean diurnal range divided by, so it's like the mean diurnal range divided by the temperature annual range. So something with temperature and elevation does seem to influence petal color. So we have this, you can consider this graph just a measure of like how important each variable is at predicting whether a flower in any particular area is the color that it is. I did not update this graph. I actually relabeled them. Class 0 is dark petals. So the dark ones, elevation's a little less important. I believe class 1 was light, and then class 2 was medium. So elevation seems kind of important for the lighter pigmented plants. But we were just kind of looking into that. We plan on incorporating soil data. But what's actually really interesting is pollen. So pollen ranges from yellow to purple. And we haven't tackled this with our fancy pipeline yet, mostly because it's like really small and inconsistent, and pollen's not always present. The anther could be present, but it might not have dehisced yet, meaning like come out of the anther. So me, my PI, and our 2 undergrads like manually shifted through like all these thousands of photos, which I said I wasn't gonna do, but I ended up doing anyway, except with help, so that was really great. So we didn't have to look at thousands each, just, you know, 1,000 each. And we ended up with about like 2,000 usable photographs that actually had like good, like a good photograph of the pollen. So like pollen was present and like the photograph was clear enough and up close enough that you could actually look at the anthers and be like, hey, yeah, like this is pink pollen. You could see like the fuzzy little pink dots here. And hey, these fuzzy purple dots, they're purple. So then I mapped that and I was like, wow, that is beautiful. I just couldn't believe that like this pattern came out. I was like, it's so nearly perfect that we've got, which is really cool. So like we originally thought like, okay, like the paler stuff, lower elevation, the darker stuff, higher elevation. And if we kind of like really snapshot the southeast, that's like exactly what the pattern is with pollen. And you remember that like most of the stuff in the higher elevation was darker petal too. But then as we like go up the Appalachians, it's that like longitude seems really important. So like where you are east-west, and it seems like you know, this coastline here is like dark pollen, but like mostly pale pink petals. And then the West Coast is like, the West area is like all yellow. And we were like, what could this be? Like, who knows? Like we plan on like looking into these questions with genetic data. So like right now we're kind of like a buzz with like, okay, like maybe it's some like historical divergence, like the Appalachian Mountains, you know, it's hard to, you know, go over them if you're a plant because you have to rely on pollinators to transport your pollen. And how are your seeds going to get over there? Your seeds don't have feet. They kind of just, you know, lay where they're yeeted. So these plants kind of just like throw their seeds out. So like you fall where you do and that's it. So it's like, maybe it's this kind of like, I guess I'm backtracking here. So like you could think like thousands of years ago, like, you know, we were covered in ice. You know, we had that glacial ice age and the species very likely like retracted south. And then as that ice melted, the species started to, you know, move back up through North America. And as it did, perhaps the Appalachian Mountains was kind of this like fence you can consider where like two clades kind of diverged. So maybe we've kind of got like this like purple, you know, colonization route and this yellow colonization route. So that's going to be really cool to look at. So right now we're just kind of thrilled that like we did actually manually shuffle through all those photographs. And then when we looked to look at what features contribute to pollen color, obviously longitude was really important for determining whether you were purple or yellow. But other really cool variables that came out had to do with like precipitation. So maybe there's something to do with like rain and determining whether you're purple or yellow pollen. So now I kind of want to like transition into like, how do we use Inaturalist and like, how is it really helpful and how is it really cool and how can I be a part of that? So I kind of just want to take a moment to talk about like good data collection techniques because after, you know, looking at so many photographs, I realized that there were a large portion of photographs that weren't very useful to me. They looked like they had been taken with a potato. So the first thing you can do is provide accurate GPS coordinates. I cannot tell you how many times I've gone to Inaturalist, I've seen like all these little dots, and I'm like, wow, that park seems great. I'm gonna go to that park and find that plant. And I get to the park and I realize that the GPS coordinate is the parking lot. And that's sad because then you don't know where in the park the thing is located, just that the person tagged the parking lot, probably as they were like getting into their car. So accurate GPS coordinates, really important. You can also put in additional information to your posting. Try to take a clear close-up shot of your observation. If it's a flower, try to get the entire reproductive structure in the photograph. That makes it a lot easier for identification purposes. And then try to get the entire organism in the shot. Of course, that's, you know, can be difficult with some organisms. If you're trying to take a picture of a really big tree, you may not be able to get like all the detail that you want, especially if the leaves are high up and you can't, you know, get a good picture of them. Don't use any fancy filters or flash photography. Then we end up with like photographs that are like overexposed and unusable. There's also additional information that Inaturalist has that you can note when you upload an observation, including phenology and sex. So I told you that I had to go through these photographs and toss out the ones that didn't have flowers. And that's because a lot of the people who upload their observations don't note that it's flowering. So you can note that it's flowering or has fruits or budding. And if people noted that, then I could have just filtered by that. But most people don't. So then I lose a lot of potentially good data that way. The sex is a different thing because they only have male and female. And I like message them, I'm like, plants can be hermaphrodites. I mean, so can some animals. So I was like, I was gonna then ask if you think there's anything that should be added to like good data collection techniques. I've seen some people include rulers in their photographs, which is actually really helpful if you're trying to like get a gauge of like how big something is.
I would say sometimes you need multiple angles. Definitely mushrooms, some insects. Yeah.
I agree. And you can upload more than one photograph to an observation. So then there are tons of positives to using this. I kind of just stole this from their website. the reasons I find iNaturalist really cool is because like you get to feel that you're like helping, I guess, like, I don't want to say greater good, but you get to be part of this like information reservoir that is going to last. Because if you look back at these herbarium specimens from like decades ago, you like, I like read the tag and I'm like, you know, reading the name and you're like, who was this person? Like, what brought them to do this? I'm like really glad they did this because we have this record and you get to be part of that. Like maybe when I'm, you know, old, my kids get to like see my iNaturalist records and like get kind of this like nature journal of my life. I don't know. It's really cool because like one, you get to learn from it. So like I like to use it for things that I'm not very knowledgeable of, like salamanders. I'm like, that thing's really cute. I have no idea what you are. And so I could just post it and be like, I don't know. And people will come and they'll be like, oh yeah, that's you know, XYZ. And you get to learn about nature. It's just like a really fun like activity for people to do when they're out in the wild. It's also really cool for me as a scientist because fieldwork is super expensive. So if there's this way that I can just like log on and get this idea of like, you know, what's out there. It's also great for like starting projects. Like, hey, I have this idea, but like, let me look into it. It's also really good for finding field sites. So like one thing I learned about using herbarium records is like, if they're really old, you have no idea if the thing is there or not. And I cannot tell you how many times I've looked at like our past graduate students' logs. And I'm talking like, this is not like more than like 6 years ago. And I go to that site and it's like, new homes for sale. I'm like, wow, that's disappointing. So elusive iNaturalist, I can be like, hey, what bloomed last year? Okay, I'm gonna go there because it's probably still there. And so that's kind of, once I started implementing that, I like had so much more success in finding like good field sites instead of just like driving up to a construction site, like where there were homes sitting. It's also great for community building. So I'll get a little bit into how you can like use iNaturalist as like a community building activity to learn more about where you live. It's long lasting and consistent. So herbariums are long lasting too, like pray to God, you hope. But things like bugs and floods in buildings can destroy samples, and that's really sad, and we don't want that to happen. But you know, thankfully here we have the protection of the internet. It's also really intuitive. You can search by year, you can search by like location, like I just want to look at Georgia, I just want to look at North America. So there's tons of filtering options, and it's community curated, so you kind of get like everybody's input. That's also a negative too. So the negatives to iNaturalist, because I know like as a scientist, I've had like a lot of these talks on like whether citizen scientist data is like actually good or if it's actually bad for the community. So I talked about having really accurate GPS coordinates, but that's actually really dangerous if the plant is threatened or endangered. iNaturalist does have this filtering system that if you post something that's, you know, endangered, it'll automatically obscure the coordinates. So it may just be like, hey, yeah, this, you know, trout lily is in Georgia, but that's all we're giving you. They do kind of have some gray area to this. So in cases where they think that the thing that's endangered isn't going to be exploited, they'll give you the coordinates. For example, this redwood tree, they're like, yeah, that thing's huge. Nobody's going to poach that, of course. And I thought like similarly, that'd be how manatees work. Because if you're familiar with manatees, they live in the Gulf. But then once temperatures in the Gulf drop, to about like 65 degrees, they like come up and swim up these rivers in Florida where it's warm, and they like live there over the winter. And I was like, yeah, they're endangered, but also everybody knows the routes they go to. Like most of these cities are like very touristy. They're like, hey, come swim with a manatee, come kayak at a manatee, look at our manatee reserves. But then they actually had the coordinates obscured. And I was like, it's a moving thing. Like if you go to the same GPS location, it's not gonna be there. So that's kind of still a gray area on what they actually obscure versus what they don't. So it is this opportunity for poaching or stealing. So usually when I reach out to parks, asking if I can like, you know, steal some of their geraniums, they're usually like, yeah, fine. And I usually mention like, hey, I'm gonna add this to iNaturalist, like, you know, add to this community curated source. And I had one occurrence where a park told me like, please don't do that. We've had an issue where people have just like been coming in and like stealing our plants, which is pretty awful. So then there's always like that worry that like somebody's like gonna use it to like find their garden, you know, like I'm gonna take this and put it in my garden. The record obviously isn't as extensive as herbarium data. You can, pop into a herbarium and probably find a specimen like decades old, like older than me for sure, but you're not going to find that on iNaturalist because it's not that old yet. But, if we keep it up, then we will have that, extensive record, which will just like, I'm so excited for it because like when I'm like old, we'll have like, like 50 years of geranium and then we can like look through it like temporally too. Like, so that'll be pretty cool. There's no physical component. So like all you have is a photograph, while with a herbarium specimen, like you actually have the thing in hand. And in some cases, you can actually take a piece of that for DNA samples. You can't do that with iNaturalist. You can walk into copyrighted images, which kind of sucks. Somebody would copyright their citizen scientist picture. It's community curated, which means people could identify the thing wrong. And there's definitely potential for human bias. So here I have a population density map of the US. So like darker orange, higher population density. And here I have my geranium map. And you notice that most of the geranium occurrences are where they're high population density. So, and it makes sense, right? Like people are at parks, they're at like places that people typically go, they take photographs, they're not going deep into the woods, deep into the unknown. So we kind of get the skewed data. So like the occurrence records are going to be skewed towards higher population density, which has its pros and cons. The pro is like, if you're in a high population density and there's like no occurrence record of the thing, it's probably not there. So you're pretty certain it's not there. However, you're not sure about the low density areas. Like it could be there and just nobody's been there, or it may not be there. So we kind of get this like skewed map of like what actually exists. How to use iNaturalist. I put this on my supplemental guide. So if you have been invigorated by this talk, you can get instructions. So I'll just really quickly run through some of the functionality. So there's an explore button, which allows you to view all observations, filter however you want. You can just filter by species or just clade, like, hey, I just want to look at all the flowers. You can upload your own photographs. Don't worry about identification. You just put in as much information as you have, and the rest will be community curated. You can look at other people's observations, and you can watch your own observations become research grade. So to become research grade, this just means that the date is accurate, it's geo-referenced. There's some evidence of it being in the wild. So like for a plant, there may be a photo. For a bird, that may be a recording of a bird call, that the community agrees on the features and they agree on the ID. So now I just want to like take a step back to just talk about like other cool projects happening with iNaturalist. So we all know about that horrible winter storm that like ripped through Texas. But there was this really cool, but sad, project started by the Texas Park and Wildlife to catalog the effects that this cold winter had on wildlife. So it's basically a project that's observation of dead animals that have been suspected to be killed from the storm. So as of 2027, there were almost 2,000 observations and 209 species, cataloged by around 450 citizens. So I wanted to ask, like, what do you think would be the most observed species? I personally thought birds, like types of birds that like would come to people's feeders, like robins.
Would it be a lizard or something?
Yeah, maybe. So actually the top 4 occurrences were all fish. Which I didn't show you the dead fish. I like found, pictures of the alive version, figuring that nobody signed up to see dead things. Which then asks this question of like, do you think this list is actually reflective of the total on species? Because what I was thinking of was like, hey, a lot of these fish, especially like catfish, are things that people like, fish for. It's a game species. So is it more so that like, these are the things more impacted or the things we're more likely to see because people go fishing, so they see this kind of thing, or, things that happen around humans, people aren't just gonna like go walking into the woods looking for dead foxes and nobody's gonna notice like a burrow of mice that have, perished. Mm-hmm. So like maybe there's that bias too. Then of course, pollinators, this is a plant talk, so we might as well talk about pollinators. I cannot even begin to tell you how many projects there are about pollinators for iNaturalist. There are general surveys, so just like states and counties and like communities have their own surveys of like what pollinators exist in our area. Other projects look for associations, trying to figure out like what pollinators go to what plants. There are even just like studies for communities to be like, hey, what comes to your backyard? You planted a pollinator garden? Like what comes to it? We want to know what's like in our urban area and how our urban area differs from the rural area around us. Like our pollinator gardens, an effective way to bring a diversity of pollinators to us. So now I was going to ask like here, who has a pollinator garden? I see there's stuff in the chat. So now I'm going to use that as a big species that are more visible. Yeah, those are gonna be, I mean, like, it's gonna be hard to see like a dead fox, like that might just have been like eaten by something. I don't know. But then, okay. Then this one I thought was really fun. It's called the name game. So this will be really fun for just like, if you're out and about and you just wanna play something fun or you wanna get your kids involved in being naturalists. So this project is all about uploading observations of things with funny names. And that's all it is to it. So you got, you know, the fluffy tit, the devil's dipstick, the sarcastic fringe head, and the pobble bonk, which I think is my favorite name on the list. And then I was like, what could you add to this list? Aren't there those birds called the blue-footed boobies? Yeah. So lots of creativity. But there are even projects that you could, do. So like people who are in teaching fields or in fields where they're facing or serving the community, there are tons of ways that you can bring the community together on projects. So here I have an example of an environmental class. It's a wildlife ecology and conservation class in Maryland. Sorry, I can't even, can't see my PowerPoint. In Maine, sorry, not Maryland. They began this project, I'm assuming they're doing it semesterly, but it began a few years ago and has over almost 4,000 observations. So basically they just task their students with like seeing like the natural history of their area. So if you have a class or some type of organization, you could do something like this, like, hey, what's around the botanical gardens? Or like, what does Athens look like? There are other community themed projects. So I actually came across this one. It's brand new. It started just last month. Actually, I think it started February 22nd. I don't know why I wrote January. Anyway, it started this year. And it's this project on UGA grounds to look at bird collisions because these people that started it must be really passionate about like, hey, like birds are flying into these windows and dying. And that's like not cool. So what you do is if you find a dead bird on campus that you suspect flew into a window, you snap a picture, upload it to iNaturalist, and they want to use this data to educate UGA about this problem and to encourage them to make buildings bird friendly. So you can actually like, there's certain decals that you can put on windows that keep birds from flying into them. So it can like stop the sad bird deaths. So I thought that was really cool. So definitely look into that if you're on campus. Other community projects include Bioblitzes. And these are just fun community events that you use just to catalog species within a specific time frame and location. So they're really good for like coursework, just being a naturalist, outdoor enthusiast, just anyone. So they can be really, really specific. So one that's running in our area right now is the NPS Cumberland Peatland Network Invasive Species. So this is running currently across like one cluster of parks. And basically what they want is like a catalog of all the invasive species in that area. And it runs all throughout this year. So if you find yourself in any of these parks, and I've given you the link in my supplemental file, you know, consider adding on to that. So, you know, let them know if you see any kudzu. Another example is just this one city, this city of Burlington just wanted to know like, hey, what's present in our area? And anyone can start this. So you could do one for like, hey, like, what flowers are in bloom, you know, in April in Athens. So go outside and take lots of pictures. You can bring a friend or go alone. And I showed you some of my observations. See the geranium, looks really familiar. And then upload them to iNaturalist. You can do it by your phone, by your computer, via your more tech-savvy friend. And there's this new app called Seek that iNaturalist hosts. So if you're not good at names or you're not sure what you're looking at, and if you want to expand your knowledge and have fun, you can use this app. What's different about it is that it uses AI to identify the species. So word of warning on that, it may or may not work well, but that is pretty cool if you just want to go outside and see what's around. So acknowledgements, of course, I have my advisor and my wonderful committee who's had to listen to this so many times. I have my husband who's been my field assistant and my partner in this research. I've got my undergraduates, Emma and Mackenzie, who have helped me a lot. I've got Kai and Amy and Ling Yang and my mom who have all come out on field work with me. Inaturalist, of course, and everybody on Inaturalist that loves geranium. Yeah, I think that went a little bit longer than I thought it would, but it was fun.
That was great. Thank you so much. Clapping on Zoom is kind of a weird part that we're working on. That's fantastic. There are a few minutes left if anybody has questions for Rachel. I know Dr. Freeman was curious about your pipeline and whether it works for other organisms yet.
Yeah, so I might tell my husband to unmute himself. That's like our goal right now, to get it to work for other flower species. What we're realizing is that white flowers are really difficult. And that's just because you get like these dead leaves on the ground, and like the sunlight hitting the leaves like kind of get back like a white color. So sometimes we get this like extraneous, you know, information into our clusters, but that is the goal. And we've been working on it with actually my undergraduates right now, who one of them's in the talk right now, Emma. She's working on it with a different geranium species. And Mackenzie's on there too. So we're trying to see how far we can get it to work.
And I was going to suggest, this is Shumei. I was going to say that, you know, when you were saying that, put a ruler next to it, you know, we've actually noticed that if the ruler's color is really close to the flower color, that becomes a problem as well. So, so there's like various, if you wanted to use this pipeline to sort of automatically, you know, get the data out of it, there are some issues that, you know, that needs to be dealt with, I guess.
Yeah, I've heard, and I think they've only rolled it out in part on the West Coast of North America, but like the sea stars I work on, there's now a way where it's either has a button or some way that it's detecting whether there's disease symptoms or not, which is really useful for similar research. You know, just people can say, hey, this sea star looks gross, and we'll go work on it. Well, are there any other questions? Rachel, this was really fun. I really appreciate it. You did a great job. And now I'll look for some geraniums in the coming month or so to add them to the data. So
post them on iNaturalist.
That's right. Well, thank you so much. For everybody who's here, remember that April 1st will be our next talk. It'll be on fireflies by Dr. Katrine Stanger-Hall. And again, that one will be really well timed to come out just a few weeks before the fireflies do. So thank you very much. Thank you, Rachel. And we'll see you next month. Cheers.
Bye. Thanks, y'all.
Bye, Rachel.
Bye, Dad.